Right, Jasmine had to go to our own lab to carry on with her research. She'll be back a bit to help me with the other projects, but I thought it would be interesting just to try and see if I can do this solo. So I already showed you how to do those 4 different types of dilutions and plate our mixture of cells onto YEPD. Now I'm going to repeat those same 4 kinds of dilutions, do them all over again, but this time I'm going to plate them on well, it's the same medium, YAPD, but it's got a drug added. The drug is called G418, and this drug allows only one of the cell types to grow. So one of the 2 cell types in the mixture is resistant to this drug. So they'll be able to grow, but the other cells will be killed. So we can use, therefore, the number of colonies on the 2 different types of media to determine the numbers of each type in the mixture overall. So yeah, I'm going to try and do this all kind of one-handed and filming with the selfie camera at the same time, and I think it'll be interesting, it'll be useful for me to see how the different filming methods compare. Now, so you don't have to watch me labeling everything again, especially one-handed, I've already prepared some of this. Let me show you. Okay, so we've got, I've already labeled the plates. So that's for the streak, we'll do that one last. That's for the 10 tiny dilutions. That's for the 5 1 in 10 dilutions. And that's for our big, one single big dilution. And I've already put beads on them. Okay, they've just fallen onto the lids now, but I'll just put them in an order. And I've already prepared the volumes for doing the dilutions. Here's a fresh, it's a new, this was the one I used before. I wrote and used on it to make sure I don't accidentally reuse that. That would be bad. So this is a fresh, sterile litre of water. Do that. Here is the same mixture. It's the same mixture that we used before. I'm going to vortex this. Switch that off. Vortex that. Put that there. Open it up. Right, I'm going to take 10 microliters. That's the that's a P20. Set at 10 already. 10 microliters of that mixture. Put it in to a litre of water. Eject the tip. Lid on. Mix that up really well. Lid off. And plate 100 microliters. So that, now you can see, I really can do these things one-handed. Okay, that's my P200 set to 100. Okay, and onto G418 medium. So the only difference, the only thing I'm doing differently this time is I'm placing them onto different media. This medium, G418, has this drug in, which only allows one of the 2 cell types in the mixture to grow. The first media I used, YPD, lets everything grow. Okay, so spread that. Okay, now I'll do the same dilutions that I did before. So first of all, the 5 1 in 10 dilutions. Okay, so I've got my pipetta set to 100 microliters. I'm going to re-vortex the cell mixture. Okay. Take 100 microliters of the mixture, pipette it into 900 microliters of sterile water. Okay, 1 in 10 dilution. Inject the tip. Use a new tip to mix it. Close the lid so I know where I've got to. check the tip, new tip, mix. It's quite interesting doing this entirely one-handed while I'm holding the phone with the other. New tip. And that's the final one. New tip. Mix. And onto the G418 plate. Okay. Again, that's the plate that only allows the resistant strain to grow. All right, we're done with those. So I'm going to throw them away. Next comes the dilution series, where we do 10 tiny dilutions of 31.6 microliters added to 68.4 microliters, so adds up to 100. And we're using the P200, which isn't ideal. It would be better if we could use a pipette with a slightly smaller volume, because 31.6 is the end of the effective range for this one. 31.6, there we go. Right, actually, I need to vortex the mixture again. Draw out 31.6 into the first dilution. Actually, okay, change tip. Into the second one, so I don't get lost. Change tip. Into the third one. Change tip. Into the fourth one. Change tip and mix. Fifth one. Change mix. Sixth one, mix into the seventh, mix with a fresh tip. It's quite zen doing these sorts of things, really. So in the eighth, mix into the ninth. Check my tip, get a new tip. And into the 10th. Right, now, I'm going to pipette all of that, and we're expecting it to be 100 microliters. And these, oh yeah, look, this is a new innovation. You can turn the actual top bit there. The really old Gilsons, you couldn't do that. Right. 100. Okay. Pipette all of that up. Okay, pretty accurate. And we'll pipette that onto the plate. Now you could see that what happened, you can see there's a bead came out, and that's because I tipped them upside down, didn't I, to show you the writing, and then some of the beads stuck to the condensation on the top. So just be a bit careful if some of them have fallen out, but that doesn't matter because there are a few on the surface. I'll clear those up and I'll sterilize the bench with some ethanol in a minute. Okay, spread those. Right, the last one is the streak, which you'll remember, I'm sure. So I need to get some sticks ready. And I realized I put beads on the streak plate, which obviously I don't want. Tap those onto the waste. Right. So vortex my mixture for the final time. Pick sterile stick. Dab it in there. And as I showed you last time, I'm gonna streak across 4 quarters using a different stick each time. All right. Now I'm going to try to pick the cells. This is really hard one-handed, but I think I can do it. Pick the cells from the end of the streak and streak them into the new corner. Change stick. Pick the cells from the end of that streak and move them into the 3rd corner. And finally, I'm going to turn the plate a bit. I can see, I don't know if you can, but I can see the where the previous streaks have been. Pick those and move them into the final corner. All right, so those are the same dilutions that we already did with the much more professional filming of Jasmine, but I've done them now again onto the different media. So we'll be able to compare the numbers of the colonies on each to determine the numbers of each type. Okay, I think that's it.